Hey, thank you again for inviting me to give a talk at your bird club. I really appreciate the invitation, and I'm sad that Jason, who I overlapped with for quite a few years at the Field Museum, is not here. But there's like multiple connections between me and Jason. So I think he got his master's degree at the University of Minnesota, which is where I am right now. Um, and I like to start my talks with a little bit about myself. And so to make somewhat of a connection to the Philadelphia area, I grew up in New York City. This is a small um, uh, concept map of me, of how I got here to the Bell Museum. I grew up in New York City, and as you can imagine, didn't get a lot of chances to be out in the wild and out in nature in the city in this very urban setting where we lived. But I did spend a lot of time coming over to Philadelphia to visit family in the suburbs of Philly, and so I spent much of my childhood around there. Um, I'm also the child of immigrants, and as you probably, many of you know, immigrant families tend to really try to push their children towards professional degrees that are things like engineering, medical doctor, lawyer, business person, things that they can easily identify as professions that will earn money. And so I was really pushed towards a career in in medicine. And it took me a long time to kind of uh, come away from that saying, I'm really interested in biology, but I'm not interested necessarily in human aspects of biology. And but I really wasn't sure what the other possibilities were. And it took me quite a while through college and towards the end of college when I finally was able to say, there's a lot of other careers in biology that don't have to deal with humans and medicine and health related careers. And so I was introduced to growing up in New York, I spent a lot of time in museums and the public side. And right before I graduated from college, I got invited to do an internship at the American Museum of Natural History. And the first time I walked behind the scenes of that museum and walked into the ornithology department, I it just felt so right. It felt like this is what I was looking for how I wanted to kind of charter my career. And so there was something about that very first visit, and that internship led me to lots of other opportunities. This is also my way of saying I came to birds very much from an academic perspective. I didn't come in necessarily being a bird watcher who wanted to study birds. I became a bird watcher after I decided to study birds in my career. And I'll tell you a little bit more about one part of my research as I continue. And since this is a fairly small group, I would be fine if you guys wanted to interrupt me with questions as I'm talking, or if you wanted to wait until the end for questions too. So I'll try to speak slowly so that I can make sure I can touch all these points, but I do have a tendency to kind of go off on tangents. So if you need me to go over something, again, I'm happy to do that. So in my research, in my lab, in my science, what I really appreciate is the fact that it allows me to explore the world through science. And I do this by really focusing on museum collections and all the things that museum collections can tell us and allow us to do. And also the people that come with these collections, right? So the people who have contributed to museum collections along the way, as well as the people that I can help get excited about science through these types of data, through these types of systems. And so in my lab, what we really do is we study evolutionary relationships of birds. And we do this using genetic data. That's what's depicted here. We also look at geographic variation and where birds are in relation to known areas of high diversity, or how do we understand diversity across landscapes. We also use this information to look at how climate and ecology might influence diversification of birds. And then we use kind of related aspects such as parasites and potential coevolution with parasites, as well as morphological evolution, both in terms of hard anatomical features as well as plumage, to look at how all the different aspects of bird diversity came to be. And I want to make sure that I give ample credit to all the people that came before me and with me in this journey. As I tell you about my studies in Madagascar, I really want to acknowledge that all of this is work where I'm standing on the shoulders of giants, right? None of it would be possible without all the people that came before me, that built up the decades of surveys and specimens and research that are now in museum collections that allow people like me to be able to use them to using modern tools like genomic tools or CT scanning or other modern tools to be able to bring new insights to these using these specimens. I also want to give a shout out to all my collaborators and my students who have worked with me, especially the Malagasy collaborators that I have worked with, including Mary-Jeanne and Steve and Manu, who are all people that have participated in field research as well as museum research that I will be talking about. And then in these panoramic photographs, you see pictures of a number of undergraduate, graduate, and postdoctoral fellows that have been mentored in my lab. And I really, I love museums. And so when I can find students who are equally fascinated by this and want to participate and want to be able to learn more about biodiversity through collections, it gets me really excited. So this is definitely one of my favorite parts of my work. And before I continue further, I also would like to just acknowledge the role the collections play and the role that I feel like I play in acknowledging that all of museum collections have a past history, and in some cases, this past history can be problematic and harmful to some cultures. And so by acknowledging this, we are also saying that we will continue to use these specimens in ways that will create new science and knowledge, but we also want to use these specimens to create a way and use museums to create an environment that welcomes and empowers people from historically underrepresented and marginalized communities so that they have equitable access to these resources. These are global resources in my mind, and we should all have access to them. But acknowledging the fact that in some cases these were collected in rather violent ways is also a step forward in for us to acknowledge that not that there's things that we can do to give back to some of the cultures that were harmed during the collection of these specimens. And one thing we can do is repatriate knowledge, repatriate the access to these specimens. So I think it's really important that we continue to use these specimens to better understand the natural world, but also acknowledge that we can do more to provide access to people who don't have it currently, so that these continue to be a global resource. So my research is on bird diversity and the evolutionary history of birds. And I have worked at multiple levels, looking at modern birds, living birds that we see today. And one thing that I think is really interesting is that birds of all of the major groups of organisms are amongst the most well-known, right? We know more about birds than we do about other vertebrate groups like fishes or mammals even. And we know more about birds than we know about plant diversity and most invertebrates. So when I say we know more about birds, what I mean is we think we've described most of the diversity out there. There's still new species being described for sure, but the rate at which new species are being described is much lower than in most groups. So we think we've kind of reached, close to the peak of knowing what's out there. But what we're also finding is that we have this kind of sense that birds are well known, but it turns out some birds are really well known, and there's still a lot we don't know about a third of birds. So of the 10,000 species, roughly, of birds out there, we have entire genomes for more than 1,000 of them. So that's a lot. If we can use that metric, genomes are a good metric of modern ways of knowing a species. And so we have entire genomes and all the information, all the genetic code of more than 1,000 species. But then there are about 3,000 species where we know we don't have any genetic data. We have no modern information, really. And so it's really biased and really divided in terms of how well we know birds. And if we think about where these gaps are, where are the species that we don't really know much about? If we map this onto a global map, what we find is that these darker areas are the areas where there are more species that have no genetic information. And we find that and this is a map that is showing the proportion of species with no genetic information. So taking all of the species that are in a particular area and mapping and highlighting the proportion that have no genetic data. So we know that the tropical regions have more diversity, but what this map is showing you is that not just the tropics, but the old world tropics, Africa, Asia, are areas where there's a disproportionately fewer number of species that have been sampled using genetics. So we know disproportionately less about these areas than we know about any other parts of the world. And for me, this is kind of my mandate. I want to make sure that we understand birds, and especially birds from these understudied areas well, so that we can begin to really ask more synthetic questions of why there's more diversity in some areas than others, and how that diversity came to be. And this is just to expand a little bit further on that. Here are maps that show species diversity across the globe. And again, we have the tropics highlighted, and then species without genetic data. Again, we have the tropics highlighted. This makes sense. There are more species here, and so there's going to be proportionally more species that are not sampled in genetic studies in the tropics. But then if we think about the proportion, the total number minus the number that are not sampled, we get a much higher distribution of them in the old world tropics. So this is where I focus most of my research, in the old world tropics, or the Eastern Hemisphere tropics. And today I'm going to focus in on one particular area in Madagascar, which is this island here off of Africa, as you I'm sure all know. And Madagascar is one of my favorite places, and one that I dreamed about a lot when I was growing up in New York City, and I spent a lot of time watching documentaries, and this is a dream come true for me, that I was able to, later in life, be able to kind of form a whole research program surrounded by my interest in this group that started first as just a hobby and some place that I just thought was fascinating. And part of why I think it's so fascinating is because Madagascar is just home to a bizarre number of critters, right? You probably are all familiar with lemurs. Lemurs are found nowhere else in the world. And there's an incredible diversity of chameleons and a whole host of other animals and plants, including my favorite group, the baobabs, that are only found in this particular island. So, and I'll tell you some more about like the bizarre birds of this island in a little bit. So we think of islands as natural laboratories, and these are areas where it's hard to get to as an organism. But if you're lucky enough to get there, you will probably experience reduced competition and reduced number of predators. And so we think of islands as these natural laboratories where we can see evolution happening often faster and in more dramatic ways than we would in continental systems. And famously, we know that we have good examples of this, of these groups like Darwin's finches and Hawaiian honey creepers, which are groups that are endemic to island systems and have really evolved to fill niches or make use of resources in ways that have led to dramatic adaptive evolution. And in Madagascar, we have a similar number of examples of adaptive radiations or endemic radiations. This includes the lemurs and the chameleons and the baobabs, as I just mentioned before. All of these are representations of lineages where there were a few individuals that were able to get to Madagascar and then really diversify into very dramatic numbers of species and that all look quite different from each other. Before I tell you more about the birds, I want to tell you a little bit more about Madagascar itself. So Madagascar, about 120 years ago, was part of the supercontinent of Gondwana, and it became an island after this supercontinent started to break up. So it was last connected to Africa about 100 million years ago, and last connected to India about 88 million years ago, and that was the last time it was connected to anything else. And Madagascar has remained isolated for the last 88 million years, and in roughly the same location. It kind of migrated a little bit north, but stayed very closely and stayed in close proximity to Africa, but has not really changed much tectonically for the past 88 million years. And 88 million years ago, this is what the birds look like, right? So this is before the Cretaceous Tertiary mass extinction event, and dinosaurs were roaming around. And we all have a number of bizarre forms of non-avian dinosaurs that we non-avian dinosaurs that we know from this region. And so Madagascar actually has some really cool examples of a host of dinosaur diversity. So we have sauropods and theropods and a whole number over here. There's a number of species that have been recently discovered that are actually quite close to the lineage that leads to modern birds. So birds are dinosaurs, and birds are the last lineage that survived the mass extinction, whereas all the non-avian dinosaurs go extinct. And so that's happening right here where this arrow is. And before that happens, when Madagascar is already an island, this is kind of what we would expect the avifauna to look like. What I want to point out here is I don't know a lot about the dinosaur diversity to speak intelligently, but what I do find fascinating is that even of the few examples that I have here on this slide, you can see that there's some really bizarre forms. Here's this guy, Mysacosaurus, that has this underbite that is unusual in dinosaurs, and then this very bird-like dinosaur that has a big bill that's just very much reminiscent of one of the examples I'll show you in a bit. So even then, we see these very unique groups and bizarre groups that are not found in other regions, and so I think that characteristic of evolving in Madagascar stays true throughout its history. So obviously, modern birds and modern animals, like the mammals that we're familiar with, many of the reptiles and other vertebrates that we're familiar with hadn't evolved when Madagascar became an island. So how did they get there? How did they come to be, and how did they get to Madagascar? Well, a famous paleontologist slash mammologist at the American Museum of Natural History looked at the patterns of mammals in Africa, and Africa has probably the highest diversity of mammals in the world, and especially of large mammals, sorry, and looked at what was in Africa and looked at what was in Madagascar and thought, it's strange that there's not a lot of overlap with what's in Africa and Madagascar, and why might that be? And so he proposed this idea of a sweepstakes model, that only a lucky few were able to win a lottery ticket that allowed them to cross the Mozambique Channel and get to Madagascar. And once they got there, they had a whole bunch of resources that they didn't have in Madagascar, in Africa, and could really diversify. Well, I think this model is really interesting and kind of interesting in multiple ways. One, I think, is it serves as a good model for the Madagascar movies, which seem to all be about how African mammals might be able to make it to Madagascar through by somehow going through New York City. It's just, I feel like the filmmakers must have seen this graphic of Simpsons and tried to create a whole movie scenario around that. But what's interesting also in terms of Simpson's idea of the sweepstakes is that, there's really a very limited number of organisms that made it from Africa to Madagascar. And this was a question that kind of confounded people for a long time. Why are there so few lineages of mammals that made it? In fact, there are only about 5 lineages, 4 land lineages of mammals and bats, which it's unclear if bats probably came multiple times to Madagascar, but they fly. So of the ones that don't fly, only 4 lineages of mammals have been able to cross the Mozambique Channel from Africa and make it to Madagascar. And from there, they, over millions of years, have diversified into 250, at least 250 species that are only found in Madagascar. So this is huge, right? This is a huge disparity that we can notice in this context. And the one explanation for this is that the wind and ocean circulation patterns were actually blowing in the wrong way for allowing the dispersal from Africa into Madagascar. So for most of the Cenozoic history, the wind circulations were actually blowing from east to west rather than from west to east. And so because it didn't favor the dispersal, we have very few lineages crossing, and only a few times when the ocean circulations reversed and allowed some of these lineages to come through. Okay, so that's the explanation for for mammals, right? So only 4 lineages that make it into Madagascar that lead to this incredible diversity. But what's going on with birds? Well, if we look at the birds that are in Madagascar and plot them on the bird tree of life, what we find is that there are actually multiple lineages across the bird tree of life. There isn't quite the same dramatic pattern that we notice with mammals. But what we do notice is there's an interesting contrast between two types of lineages that have been able to get to Madagascar. What we see is that there's some lineages that got to Madagascar and were able to diversify once they got there. So they were able to speciate in situ and create new species in Madagascar. But then there's an equal number of species that get to Madagascar and never diversify. And this sets up a really nice contrast for us to test why this might be, right? Why are there some groups that diversify more than others? And then the opposite, why are there some groups that just fail to diversify, right? And can we use this to predict what might cause groups to diversify? What is it about either the environment or other aspects of diversification that might lead to this? And is there something we can get from the system that might help us to predict patterns of diversification and rates of diversification? And so this has been really how I've been thinking about my work in Madagascar. And what I'll show you in the next couple of slides is how we've started to approach that. And this is an ongoing process. And so we're able to answer some questions, and we're still working on getting a full picture for a lot of the other questions. So the first question we asked was, how did birds diversify within Madagascar? And we were looking at that contrast between groups that were able to diversify once they got to Madagascar versus ones that did not. And we wanted to see if maybe there was some sort of phylogenetic aspect to this, right? So there are some groups that have a higher speciation rate. They just naturally can speciate faster. And so is there some aspect of the speciation rate of a particular group that is what is driving some groups in Madagascar to be able to speciate more? And what we found is here in this plot, we plotted speciation rate versus the total number of species of any given group in Madagascar. And we find that there actually isn't a good correlation there, right? There's a number of lineages that have both low speciation rates and particularly high speciation rates, but on Madagascar, they just haven't speciated. And so there's no real correlation there. And what we find is that the groups that have the highest diversity tend to be the ones that have the lowest global speciation rate, so speciation rate across the world. And to highlight a few of these, the largest radiation on Madagascar, the largest bird radiation on Madagascar are the vangas. And these are exemplified here in these pictures and this group. And this was a group that not until the year 2012 were we even sure was a monophyletic group, that they shared a common ancestor. And all the species that have stars here are species that were newly put into this family. These were species that for a long time were thought to be parts of other families, very distantly related families. And only with a lot of genetic data were we able to show that these are actually part of the Vanga clade. And what's extra cool about this, and you'll see more examples of this later, is that this actually expands the breadth of this radiation even more in terms of how ecologically and morphologically diverse this group is. And then just again to highlight how our knowledge was so limited, even just 10 years ago, the second most diverse group in Madagascar was a family that we only discovered in the year 2000, and only with the use of DNA. So all of these all of these birds that are in this clade here in this group were originally put into families like warblers, old world warblers, babblers, and bulbuls. And it turns out that there are no warblers, babblers, or bulbuls in Madagascar. All of these actually belong in this endemic radiation, and they have a lot of similarities to warblers, babblers, and bulbuls that are found in Asia and Africa. And so for a long time, taxonomists had assumed that that's where they came from, but it turns out they had independently evolved to be very similar and to do very similar things like warblers and babblers and bulbuls. So, the power of being able to re-examine specimens, re-examine the natural history of organisms, and really figure out what's going on is so important in these contexts, because a lot of our assumptions from the past can be tested using more data, and so that's what's really what drives me when I'm doing my research. I'm just gonna walk through a little bit more of like just what the bird diversity in Madagascar is, and when we think these birds came to Madagascar or evolved in Madagascar. So on the top here is a timeline, and here around 55 million years ago, this is after the mass extinction event that happens here. We have the first paleognates. So these are birds that are related to ostriches and emus. And in the bird tree, this is kind of the first splitting off of a group of birds from all the rest of birds. And in Madagascar, we have the largest modern bird that ever lived, the elephant bird. And this bird was about 10 feet tall. And here's an ostrich for scale and a human for scale. And what's also exciting about elephant birds is that they laid the largest egg that's physically possible for a hard-shelled egg. And I like to push this statistic because I think everyone assumes that dinosaurs laid the biggest eggs, and well, birds are dinosaurs, but birds are the ones that laid the biggest eggs. And you have David Attenborough here for scale to see how big this is. It's at least 10 times bigger than a ostrich egg. So for a long time, these birds, these ratites, we thought of them as diversifying during when Gondwana broke up. Right, we have representatives of these large flightless birds in most of the major land masses that used to be part of Gondwana. And so the classic story of biogeography was that when Gondwana split apart, you have ostriches that are in Africa, rheas that are in South America, emus in Australia, cassowaries, and that's how it's split apart. But we know that this lineage evolved a lot more recently than the breakup of Gondwana, and so we can't use that explanation. And it turns out that when we look at the phylogeny of these paleognates, it doesn't quite match that story of Gondwana breakup. And so here you should be looking at C. And I've circled here where in the traditional view, which is what B is, where the elephant birds are. And it turns out, we used to think that they were closely related to ostriches given their size and their location because Africa, Madagascar are close by. And it turns out that elephant birds are actually most closely related to kiwis, which are only found in New Zealand on the other side of the world here. This is Madagascar, this is kiwis. And so this again gives us more support for this idea that the diversification of ratites is not necessarily tied to Gondwana, but actually might have a lot more dispersal across oceanic oceans that and that probably played a role. And so you end up having a pretty disparate set of species, elephant birds being the largest, kiwis being the smallest, being most closely related, but they share other characteristics, whereas kiwis are, we think that kiwis, well, we know kiwis are nocturnal, and we think that elephant birds were also nocturnal, and they share a fair number of morphological features together. So I think it's a cool natural history story. A few around the same time, we also have a few other lineages of Madagascar. And what's cool here is that there are these two lineages, the Mezites and the cuckoo roller. Here is a kind of the base of a tree of life of birds. Oops, if you can see this. So if these are all the major lineages, and Mezites and cuckoo rollers are, I'm gonna try to show that, are these relictual lineages. They don't have any close relatives that are living today, and they are kind of as old as all of the lineages of birds that originally diversified after the Cretaceous Paleogene. And so it's amazing that we have these lineages that have just kind of stuck around. We don't think they've diversified much. We don't think there's a lot of hidden or missing diversity that has gone extinct. We think that these are lineages that, kind of like a lot of other island lineages, there's a limit to how much you can diversify on an island. And so these are kind of evolutionary dead ends in terms of being able to lead to more lineages, but interestingly enough, got here early and stayed very similar to what they evolved to be shortly after all birds diversified. So these have no close relatives in living today, the Mezites and the cuckoo rollers, and they are endemic orders to Madagascar. Around 40 million years ago, we have ascites, which are suboceans, and ground rollers, which are related to other crassiforms. And so these are groups that we have that have diversified a bit. There are about 4 or 5 species in each of these clades. And you can see that they also have some similarities to other well-known groups like sunbirds. So this one's called a false sunbird. That's not closely related to sunbirds. And so you have, again, these kind of adaptations to morphologies that are repeatedly seen in multiple groups, such as this curved bill. And then about 20 to 15 million years ago is when we see those 3 largest clades that I mentioned in the first slide, when I plotted the number of species in each group. So vangas, tetrakas, which is the or Bernierids, which are the second most diverse group in Madagascar, and kuas, which is the third most diverse group. And so these are the ones that have the largest number of species in each of these lineages, and they got to Madagascar around 20 to 15 million years ago. And then finally, we have a whole number of species that are that came to Madagascar very recently, in the last less than 5 million years ago, maybe 3 million years ago, in the Pliocene and Pleistocene. And most of these are species that are represented by those single lineages, right? Ones that have failed to diversify and haven't and are just single lineages represented in Madagascar. So we wanted to ask the question of how did birds get to Madagascar, right? And the first question, the one way to look at this is to look at where their close relatives are. And we can use that to say, well, if their close relatives are in Africa, it's most likely, given any other evidence, that they came from Africa. They dispersed from Africa. And so looking at where Madagascar is, we tested this with looking at all of the areas that are surrounding Madagascar. We tested Africa, Asia that's further away, and it's harder to see this on this map, but there are a number of small islands along the Indian Ocean that might have also aided some dispersal across the Indian Ocean coming from Africa, coming from Asia. And so if we looked at all the lineages of that are now in Madagascar and looked at where their close relatives are, it turns out that only about a third of them are coming from Africa. And we would have expected a lot more of them to come from the area that is closest to Madagascar. But it turns out only a third of them are from there. And a third of them are coming from Asia, which is a lot further away. And then a third of them we're just not sure about because either like the Cucurola and Mizites, they're just too old and don't have any close relatives, or we haven't worked out their phylogeny yet to really know how they came to be. So we think this is an interesting pattern, and it kind of defies some of the null expectations that we have in island biogeography, which kind of predicts that the closest landmass is where most of the species that are on an island will come from. And that's not what's going on at all in Madagascar. Right. And then we also looked at whether timing might play a role in where they came from. So I mentioned before that the ocean circulation and wind circulation was blowing in different directions during this time when birds were coming into Madagascar. And so for about halfway from the Paleogene into the Miocene, the wind was blowing from west to east. And then about halfway into the Miocene, the pattern switches and the pattern goes from east to west. And so when we look at that and when we look at the origins of these species, what we find is that the older lineages, again, we have a fair number of lineages that are coming from Asia, as we might predict, because that's where the sorry, more are coming from Asia, which is different from what we would predict, even though that the wind is blowing from Africa. And the opposite is true in terms of more recent times, where we should predict that more things are coming from Asia, but we end up with a signal where there's a lot of species coming from Africa. And so it doesn't quite match up with the predictions based on paleo environment. And then we also looked at whether timing was related to whether they could diversify or not. And, we think that if lineages can get to a place earlier, they have more time to radiate. And so we should expect that if they've been there longer, they have had a chance to radiate. And so that is the pattern we see. More of the lineages that got to Madagascar earlier are the ones that were able to diversify and speciate within Madagascar. But what's interesting is we still see, a little less than half that have failed to diversify. And so it's not quite a clear-cut pattern as our expectations would be. But we do see that a lot of the recent species that have gotten to Madagascar have not had the time to radiate. And so that fits our pattern, that fits our expectation. So, I mentioned a few thing to show that Madagascar is kind of a unique island. It doesn't quite it doesn't quite follow all the predictions of what we would expect based on island biogeography theory. And it also behaves a little bit like a mini continent. It has a lot of diversification. It has a lot of diverse habitat areas and areas where species can be exposed to different types of environments and ecosystems, and this might drive some of the diversification that's happening within Madagascar. So one of the predictions in island biogeography is that the size of an island is an indication of the amount of diversity it can support. And on this graph here, what it's showing is it's plotting island area versus the number of species that are found there. These are bird species that are breed on it. And this is kind of a well-known relationship in terms of area and number of species. But what we see is Madagascar is kind of off the chart, right? It's not along the lines at all. It doesn't follow that relationship. And there are a lot fewer species on Madagascar than we would expect given its area. And we asked, why is that? What's going on here? Well, one possibility is that there's cryptic diversity. So there's a potential that there's more species than we have seen or described. And if you remember from my earlier slides, I told you that we only recently discovered some of these families on Madagascar. And so if that's the case, it's possible that we're missing some species. And a good example of this is mouse lemurs, where in 1992, there were 2 known species of mouse lemurs. And then over the last 30 years of being able to do surveys and use genetic data, we've been able, that number has increased from 2 to 24. So a really dramatic increase in species. And in birds, when we first started this, we weren't really sure what to expect. We weren't sure if there might be potential for hidden species that morphologically look similar across their range, but have a lot of genetic variation. And the patterns that we had from previous studies kind of indicated the full gamut. So there were some clades that had really deep divergences, even across species where they were practically identical looking, but in terms of genetic variation were really deeply divergent, as well as in other cases where there was just no variation at all across the entire island. So, excuse me. So we began to look at this by sampling across different species and looking at populations in all the different biogeographic, bioclimatic zones, and built up a number of phylogeographic patterns to see if there was any kind of signal, anything that we could find in common that would help us to recognize some of these biogeographic patterns. What we found so far is interesting in that there isn't a single pattern, but rather multiple patterns that we're seeing. So we are seeing, in some cases, there's a split between populations on the east versus west. The eastern mountains here are really humid and wet, whereas the western parts are really dry forest, and it seems like species have been able to diversify differently in these areas. There's a north-south division in some groups, and in some cases, this where that divide happens can be quite different. And in some cases, we actually see all of that. So we see a east and a west and a north and a south. And in some cases, there's an extra lineage that seems to be found all around, but doesn't seem to be mixing with the populations that are different on the east versus the west. So a lot of complex patterns that we're uncovering as we look at whether there's patterns of diversity within Madagascar, within these species. But what we have found is that there are a number of undescribed species that were cryptic, that morphologically don't morphologically look very similar, but genetically are quite different, which indicates that they have had different evolutionary trajectories. And then once we've had that genetic information, we've actually gone back to museum specimens and looked for whether there is some morphological features that we can use to differentiate them. And in most cases, there are. So we've been able to use kind of an integrative approach to look at multiple ways of looking at speciation, looking at traits of species. And in just these 2 groups, we've already described 8 new species. So we're starting to really we're starting to kind of address that issue of, is there are there species that we are missing from Madagascar and missing from our understanding of the diversity of this region. And also just to kind of highlight why this is so important is this is probably the part that's going to be most important when we're talking about conservation, right? Just knowing how many species are where are there and where they are, and how restricted they might be to certain areas is going to be really crucial for us when we're talking about or when we're making recommendations for conservation priorities. So I think I'm running out of time, but I'm going to rush over to get to some of the some other key findings that we have here. I talked about adaptive radiations and looking at these unique features and how they came to be. And just like Darwin's finches and Hawaiian honeycreepers, the vangas have really they really show these dramatic shapes and sizes. As you can see here, there's this one here is called the nuthatch vanga, and it behaves very much like the nuthatches that we have in North America. This one here is the sickle-billed vanga, and you can see it has this very curved bill that it uses to probe into bark. And we think that they filled a niche that woodpeckers would have filled. There are no woodpeckers in Madagascar, and so the sickle-bill fills the niche of being able to eat grubs and invertebrates from the barks of trees. And so we have a number of these kind of interesting natural history stories that are related to these species, and each of these are these species are doing unique things on Madagascar than what they are doing in what their close relatives are doing in other parts of the world. And so we think of this as a good example of an adaptive radiation, but we also would like to test this using additional information and kind of test also how that can be. How do species that evolve from groups that might not be as morphologically diverse get to be as diverse as they are? And in some cases, they might take the kind of intrinsic diversity that's in that they inherited from their ancestors and just kind of expand further into that. And in other cases, they might have gone in a completely different direction than their close relatives. And that's what you're seeing here in this blue area here. The Hawaiian honeycreepers kind of diversified by doing something completely different than their common ancestors, whereas Darwin's finches just occupy more of the same space, the morphometric space that their ancestors do. So they're just able to take advantage of it in a different way, whereas Hawaiian honeycreepers are just going, we can do things completely differently. So we wanted to ask where vangas fell in that, and here's where a lot of the students that I was able to take into collections came to play a role. They helped me measure things and characterize the shape. And what we find here is when we plot the variation in their bills and in their sizes, we find that what's really interesting is here in red and in orange are the close relatives of the Malagasy vangas that are found in Asia and Africa, and green are the ones that are found in Madagascar. And you can see that the green extends the entire graph, and you also end up with like these really crazy outliers. This is good evidence that the Malagasy vangas are shifting into a different space, just like the Hawaiian honeycreepers. And they seem to be doing things quite differently than their close relatives on other islands. Other land masses. And we've been able to do this in multiple ways. We've also used photogrammetry, which is a way of taking photographs and stitching them together in 3D models. And we find the same kind of pattern. I have a student now who's using CT scans, and again, we are, approaching this in multiple ways, but finding very, very similar patterns that showed that bangas in Madagascar were able to diversify, were able to evolve in ways that they weren't able to at all in other parts of the world. They're a lot more constrained by the resources we hypothesize. And so in Madagascar, when they're freed of these constraints, you can have bill shapes like this that evolve. These are some of the things that are from our CT scans. And here's the sickle bill vanga again, where you see this incredibly thin curved bill. This bill is so thin that when we were originally trying out different types of scanners, it couldn't decipher that as being three-dimensional. It's really like barely like a millimeter wide. And then the other one here, the helmet vanga, is just such a big, thick bill that again, it's remarkable that all of these are closely related and all share a common ancestor. I'm gonna skip on to oh, one thing cool I wanted to talk about with the tetrakas and the Berniards before I kind of wrap this up is, this was the clade, the group that we only discovered in the year 2000 using DNA. And it turns out not only is it like an endemic radiation that we didn't know about, but it turns out their close relatives were really hard to identify for a long time. And we finally were able to identify it. It turns out they're really closely related to Donacobius, which some of you bird watchers might know as a bird that only exists in South America, Central and South America, in swampy regions. And kind of bizarre that they're the most closely related to a family in Madagascar on the other side of Africa. So again, we're uncovering some really interesting biogeographic patterns. And, as we continue with our research, as we build on this knowledge base, we can start to kind of piece together what's going on, not just in Madagascar, but across birds and across the world as we think about what the conditions were like and what's going on and how they came to be. So I think I will end there. And I just, want to give a shout out to just some of the things that we've been able to do so far and things we want to do going forward. We've been able to characterize some of the interesting patterns in terms of genomics, in terms of phenotypic variation and geographic variation, but we're also trying to build on this by continuing to support Malagasy scientists by giving them access to some of the specimens that are in Western museums, as well as giving them, helping them to create new knowledge based on collections there, based on field surveys and ways of kind of combining things that we can do in the US versus things that we can do together in Madagascar. And so all of this we are using to build our knowledge about biodiversity and think about what the needs of biodiversity are as climate and habitat continue to change in these areas. And then I just will end with acknowledging all the people that have helped me with the work that I presented here. And also, I'd like to dedicate all of my talks on Madagascar to a former graduate student of mine, Lenika Strozier, who we lost in 2020 to COVID. She was a really amazing and incredible person who did her master's thesis work on one of the groups of tetrakas that I showed you. Sorry, not tetrakas, vangas, the Newtonia. And she had just started her job, and when COVID hit, and just started a job in a university, and that was her dream job in a university when COVID hit. And shortly after, she unfortunately succumbed to the illness, and it's a real shame. And so for me, I think it's really important to acknowledge all the people that have been a part of this work, but also use this as an opportunity to say that there's still so much more we have to do, so much more we have to do in terms of supporting, mentoring, and really fostering people, especially people who come from backgrounds that have not had access to be able to do to have access to education and have access to STEM careers. And so I hope that you know, we all will do our parts. I continue to try to do what I can. And, you know, one thing that I do, one thing I commit to is all of the places that I go to, I talk about Linica, and all of the honoraria I get from any talks I give, I donate towards Black ornithologists so that we can continue to support them as because they are really, really underrepresented in our field. Okay, with that, I will stop talking and save some time for questions.
Thank you so much, Dr. Reddy. That was an absolutely fascinating talk. This is one of my favorite topics, evolution and species diversification. So I'm really happy to have you here and talk about that. Unlike you, as an immigrant, I became an engineer. So it's very nice to hear this talk, and I'm ready to take questions. Any questions in the room? I'd see Art.
Yes. First of all, thank you for that wonderful talk. Lots of information there and lots to think about. You showed a curve of species richness versus island size, and that's the island biogeography theory. And you showed that Madagascar fell well below that line. You show evidence that there might be a number of cryptic species in Madagascar which could move Madagascar closer to the line. Might there also be cryptic species in other islands that generated that line? So the line then might be actually moving further away from Madagascar?
Yeah, that's possible, for sure. So we do, yeah, so the island biogeography model, I'll admit my bias here. I don't think it's, I think it's a little bit incomplete, right? It doesn't take into account that species can diversify within islands, for instance. And we are developing new models that do take that into account, but it's kind of hard to incorporate all of that. But I think you're absolutely right. A lot of the islands, especially tropical islands, have not been well surveyed, and we are finding new species in these other, these other regions. And the same is true for continental regions. Continental regions in Africa and Asia. So, you know, these graphs will definitely change over time, but maybe in the same proportions. So that's a good question. Let's see what happens in a couple of years.
Murray, yes.
If I could follow up, you may have addressed this a little bit and I might have missed it, but I mean, it's part of the equation here that I think the island theory is talking about area, but of course an island is three-dimensional, not two-dimensional. So is altitude a factor there that allows more diversity, or is that already accounted for?
They don't. It's a pretty simplistic model for sure. And you know, they're counting species as all being the same as well, which as an evolutionary biologist, I kind of take a little bit you know, I don't care for that. You know, species are not independent units. They share a lot of their features from their common ancestors, and so you know we have to think about that too. So that island biogeography doesn't take into account phylogeny. It doesn't take into account elevation, as you're saying, the heterogeneity of areas, and so that's another factor for sure that we need to build into these models.
Just to follow up again on that, that graph, was that sorry if I missed it. Like, was that birds? Was that vertebrates?
There's
Was
birds.
that but is there a similar thing for mammals or reptiles or even plants? Do you know?
Yes, I believe they look similar. MacArthur and Wilson started their work with birds, but also plants. And so I think there should be like very similar models for that. But you know, in the last couple of decades, we've been kind of people have been trying to modify these models to incorporate a lot more information than the original models did, for sure.
Thank you. Any other questions in the room? Yes, Steve.
I was just wondering how much of your genomic data is based on, I mean, museum data, and how much of field studies? You know,
Yeah.
and also how much do you feel confident with your models based on the fact that a lot of the nodes of your trees, you don't really know how many there are, how many more there might be. So I'm just a little bit curious what you think about that.
Yeah, no, that's a great question. So, you know, I mentioned my colleagues in the beginning of the talk, and I really have to give a lot of credit to my colleagues who have, especially Steve Goodman and Mary-Jeanne Rahalelo, who are the two people that have led a lot of the modern surveys. And so we actually have genetic samples for almost all of the all the ones that I've been talking about. For in a few cases, we don't have any genetics tissue samples, so we have had to use museum skins. So there are a couple of species that are only described by 1 or 2 museum specimens, and they're just really hard to be able to trap in a mist net because they're kind of canopy flyers. And so for those, we've had to resort to museum specimens. So, but for, you know, we've been really fortunate for over the last 30 years, my colleagues have been able to survey all the different parts of Madagascar, well, the more accessible parts of Madagascar. There's still some areas that we're hoping to get to, still some that we that are just Madagascar is just it's oftentimes really hard to go to some places because they're just not really good roads or even roads at all. The wet zones can get really hard to cross and hard to navigate when you're either walking or driving. And so there are just some areas that are just really hard to survey. But we've been really fortunate that they've collected enough of us enough samples for us to really be able to do this work using all of the modern tools that we can. So and then the other question you had, which I think is a really good question, is like, you know, how confident are we given like how short our nodes are? Those short nodes are, and actually like part of why it's been hard to figure out the relationships of these species, because they did diversify so fast. And we see this pattern in all birds as well as in these adaptive radiations like vangas, where you know, you have a initial lineage that gets to Madagascar, and then within a million years of, you know, within a very short time, less than a million years, they have like diversified into all of the different species that we see today. And so you have a very short window that you can capture of how they diversified. And sometimes we might not be able to like fully be able to say this is exactly how it happened with a lot of confidence, just because the genetic data just won't be able to have that power. But we're finding that with all birds also, because the same happened when modern birds, you know, dinosaurs die off at the KPG, and then you suddenly have like this one lineage that becomes all the birds we see today. And and we think that, you know, within less than 2 million years or 10 million years, more than 65 million years ago is when like all the major lineages of birds evolved, right? All of them appeared, and then they go on to diversify further. But we had a very hard time kind of figuring out the pattern of how these different lineages came to be and how they evolved, because it happened so rapidly. And so we do have to accept that there's going to be some limitations of what we can do, but because that's how they came to be.
I'm going to come back to you, Art. I'm going to take a couple questions from Zoom. So Richard Weinstein asks, how do you estimate the point in time in millions of years ago that the ancestors of current species arrived in Madagascar?
That's a good question. So we use something that we call the molecular clock. So we can estimate that we think that, DNA kind of very generally, DNA evolves at a certain rate in species. And so we've estimated what that rate can be in birds. There's a lot of controversy about this because it's not an observable rate, right? It's a rate that we've inferred based on other things. And so actually using information from birds in Hawaii, we've been able to say, given the age of an island and when we estimated, a bird can only get there after the island appears. And so we can take the age of the island and the age of what we think the bird is based on the island, and then estimate the amount of genetic differences between species on different islands. We've estimated this rate of how of bird genetics evolution. And then we can apply this rate to other groups too. And so that's largely how we do it. Unfortunately, in birds, there's just not a lot of fossils for us to be able to kind of say this, this fossil fits in the phylogenetic tree over here, and therefore it gives us a sense of the range of ages for certain divergences. So in the absence of fossils, we use these estimated ranges, but I will say that there's a lot of caveats about whether these are accurate or not.
Well, thank you. One more question on Zoom. Patrick has a comment first. I love learning about island endemics. Such a great presentation. Thank you. And then he follows up with, have you studied endemics on any of the other islands around Africa?
I haven't yet, but I'm really excited to do so. There are a couple of groups that are found in the Comores and Seychelles that are kind of like surrounding Madagascar, and it would be really interesting to look at the dynamics of the groups that are found only in Madagascar versus groups that are found in all these islands versus things that have close relatives more broadly. So yeah, I think it's really interesting question.
Thank you. Art?
Yeah, thank you. You made a comment earlier in your talk that species diversity on Madagascar does not correlate with speciation rate of the source clade. Do you think that speciation, well, I guess this is a rhetorical question, would the speciation rate of the source clade, is that something intrinsic to that clade rather than just the result of the different ecological niches available to members of that clade in other environments or in other places outside of Madagascar?
Yeah, that's a good question, and we debate that actually. I mean, there is some aspect of intrinsic properties in terms of like how certain groups can speciate, or like what their mode of speciation tends to be. So for example, like I'm going to use a Eastern Hemisphere example. I don't have a good one from the Northern Hemisphere, but the white-eyes are birds that, every time they get to a different island, they speciate. But they don't necessarily speciate within the same landmass that easily, right? And so that does seem to be a loosely some sort of intrinsic way that they behave. But you're right. It's that that speciation rate is is really just thinking about what are clades doing globally and saying, okay, this is what they're doing globally. What does that mean on this particular landmass? And so it's just trying to contrast that.
Okay, last call for questions. Any more questions? Anything on Zoom? I cannot see if people have their hands up on Zoom. So if you do, please unmute yourself. It's your last chance.
I want to know what Steve meant by roads.
I think that's when you were talking about the access in Madagascar.
So we were in Madagascar in November, and I want to confirm. So what we had is our tour company sent out a thing because all of their trips had problems with the roads. And they led off the letter by saying the roads in Madagascar are somewhere between the metaphorical and the metaphysical. So that's what that is.
I love it. I'm going to use that. That's great. Did you have a good trip?
The dearth of roads.
It was both awesome and awful.
Oh,
It was, awesome in what we saw, all the lemurs, the birds, the plant life and everything. It was awful in how poor the island is, the situation, the roads. Everyone on the tour got sick, and it lasted till after we got back. But it was a magical place. It really was.
yeah. And I hear that a lot from people. I think because I spent a lot of time in India, I've like gotten used to the poor in other countries, and I think if you're not used to it, going to Madagascar can be really shocking.
Yeah, it was. I've been to other places in Africa and South America, and this place, just the houses made of sticks, right? Just single room houses made of sticks was really, really, disheartening.
Yeah. I don't know if you noticed also that there's a lot of mining for gems and semi-precious gems, and that
We
becomes
did not see that. We didn't. No, I didn't notice that. No.
Yeah.
We did notice, we went to one place where they said, yes, we're going to go there. We're going to look for this one bird that the guide had never seen. And we got it. I forget the name of the bird, but it was this little bird that was really cool that looked like another bird that we saw first, and we're confused. But anyway, he said, yeah, we'll get there and we'll have to walk maybe 10, 15 yards into the forest. And we got there and the forest was gone. And it was a national park. And the people had just cut it down to use as fuel because they have nothing else.
Yeah.
Right. And we wound up walking 1.5 miles to find this thing, but yeah, we got it. But yeah, it was that, a lot of things like that were disheartening. And, a lot of the infrastructure and the guides just told us how we were there, and the guide was there and his daughter's learning to be a guide. And we were all talking in the van, where do we want to go next? And she said, I'd love to go to Africa to see giraffes. And the fact that she could not make enough money there to afford the trip to go, across the strait, right? And she has a good job, right?
Yeah.
So, yeah.
Yeah. No, it's really disheartening.
Follow-
Go ahead.
Following up on that, are you aware, I know it's not your, central focus, but any international efforts to try to conserve, preserve, debt for nature swap model, or just anything? Here's a world treasure, as you were just, pretty much your whole talk about this remarkable place that's unique, and it's just, economically a disaster.
Yeah, no, it is a global treasure. I stand by that. I think, all of biodiversity is a global treasure, and we should care about it whether we live next to it or far away from it. And we should be putting all of our efforts in towards in whichever way we can, whether it's the local or the global efforts. The Madagascar gets a lot of, well, maybe I shouldn't say a lot, but it does get a decent amount of aid from conservation groups and aid for human development. But some of that can be really misused, and that's what I've been finding and what my colleagues tell me about a lot. So, there's a lot of funding from international groups to set up parks, and which is great, which is what we need. But if it's not managed properly, you end up with a lot of people who can take that money and use it for other things, and then there's no park, right? So it's not quite the same level of corruption that you would see in, some really well-known places. I don't want to say that while this is recording, but it's definitely, there's certain groups, just like in every society, there's certain groups that have a lot of access to power, and so they end up the ones getting benefits from this. And so, as much aid as goes into the country, it's not necessarily getting to the people who need it the most or the areas that need it the most. And that's something that needs a little bit more effort than just, sending aid or sending people there. I don't know the solution for that. I don't know how we can work towards that more. But, from the perspective of what I think I can do, it's training people, training scientists in Madagascar and empowering them to keep doing what they want to do. Because at the end of the day, it has to be that, Malagasy people are the ones that are pushing for this. We can support them, but they have to be the ones doing it.
Thanks for the really just terrific talk. Appreciate it.
I appreciate your attention. Thank you all for inviting me again. I really enjoyed this. It was fun.
Thank you so much, Dr. Reddy. Appreciate it. Thanks for spending your time with us this evening.
And take care, everyone. If you have other questions, you're welcome to reach out to me. And it was really nice to hear your meeting as well. I don't get a chance to do that much.
I do have one more question.
Yes.
Where are you that it's always 5 o'clock?
Right. It's in my daughter's room. I don't think she's like changed the battery there.
Thank you very much.
You're welcome. Take care.
Thank you. Have a good night.
Thank you. Bye.
Bye.